Okay. It's just that with the Socrative questions, I'm just wanting I'm leaving it up to you guys, right? To ask me if you have questions on the 40 questions. All right. So you have access to it. You can do it. You get the answers. We can talk about it on Friday. If you want to talk about it on Friday, I'm cool with it. We can talk about it on Thursday if you want to talk about it on Thursday. If you want to just shoot me a message on WhatsApp and be like, hey, I don't understand this question or these questions. You give me all the questions you don't understand. Then we can sit down and we can do a video chat on it. And I can talk to you through all of them real quickly. So you can have a good understanding of what's going on. Um, so either way you want to do it, I'm cool, but I'm leaving it up to you to ask me. Okay. The questions are there. You go through them. If you miss a question, you don't understand it, get with me. Okay. Good. All right. And that's going to be up until it's going to be up until tomorrow night at 12 o'clock. And then I'm going to post the Socrative for microbio, which will be for Thursdays. Okay. Good. Yeah, most definitely. Nikki's trying to get in. Yeah, I don't have anyone on my end trying to get in. I wonder. Hmm. Because I would see them on my thing. Yeah, give her the link if there's issues, please. Because I know sometimes people lose the link because you guys signed up for all the days like way ahead, right? Um, so I understand sometimes you guys lose the link, which is fine. If you end up losing the link and you want to attend live, you can just send me a message and I'll send you the link because I have my phone here with me at all times to kind of like look over if there's any WhatsApp messages that I need to address pertaining to the class, especially during the class time. I'll look through it to make sure that none of you guys have issues getting in. Okay, good. Good, good. Thank you, thank you. All right then. So now that we did that and we talked about that, we talked about what this week looks like, right? We're gonna do microbio on Thursday, MCQ. You have the end of this Tuesday to finish off the 40 questions. And then Friday, we meet up at 5 to go over the rest of Path MCQ. And we're going to also talk a little bit about the types of necrosises. We already kind of touched on them, but I just want to go through the PowerPoint just to say I've gone through it. But yeah, so those are the things. All right, so let's get started so we don't lose any more time. So yes, with microbio, a lot of it is just knowing the details. Okay. Not only knowing the details, but knowing the antibiotics or antivirals or antifungals, antiparasitic drugs. This is where a lot of this kind of bulk of microbiome is going to be. The bulk of it's going to be antibiotics. I would say about 60% of it, 60 to 70% is going to be how do you treat them. 30% is going to be what bug is it. Okay. So you really have to understand how the drugs work, what are the nuances of the drugs, right? What's the side effects of the drugs, as well as what's their mechanism of action for the drug. Those are the big things that they're going to test with the drugs. Okay. Yeah, you're going to need to know certain names and stuff like that, which you'll get with doing Anki or you'll get by constantly reviewing the material. Um, but a bulk of it is more mechanism of action. The bulk of it is more side effects. And you'll see that when we go into it on Thursday. All right. So there's Essentially, when we look at it, there's breakdown from eukaryotes, which are humans, right? You got prokaryotes, you got viruses. These are the breakdowns that we have. Now, in eukaryotes, you got to know how eukaryotes differ from the other two. The reason why I say this is because this is how we target the other guys when we look at antivirals, when we look at antibiotics, when we look at antifungals. This is how we do it because it's different than our own normal cells. Okay? For instance, between eukaryotes and prokaryotes, the big thing here is how ribosomes are different. What's the difference between ribosomes with eukaryotes? Do you know the ribosome for eukaryotes? Yes, it's a 40S and a 60S. Essentially 80, right? And then you look at prokaryotes, and theirs is different, right? They have a 50S and a 30S, essentially being a 70S. Okay, so this is the main differences with ribosomes. So does that mean that we target this when it comes to antibiotics? Exactly. Since they're different between the two of them, we target it. Okay. Another thing, the cell wall. Now, in prokaryotes, they have a cell wall. Do eukaryotes have a cell wall? No, essentially they don't. In some cells, you may say, well, what about you're looking at certain muscle cells and stuff like that? Like with the outer layers to the cells pertaining to the paramysium and stuff like that. That's not really a cell wall, right? But the cell wall is essentially with prokaryotes, and we target this. That's an antibiotic. Same thing with the cell membranes. Is another thing we target. However, in cell membranes, eukaryotes also have cell membranes. So here you have a lot of similarities when it comes to the cell membrane. Okay? But the cell wall is strictly prokaryotic. Okay? Now, the next thing is this. If there's a lot of overlapping, between eukaryotes and prokaryotes, or eukaryote and viruses, and you are targeting that with a drug, do you think you will have high side effects or low side effects? High. Perfect. So whenever you target something that is not specific for whatever you're going after, you have high side effects. I will repeat that one more time. Whenever you are targeting something with a drug that's not very specific for that one target you're looking for, and that one target you're looking for is only on the one thing this patient has being the issue, then you have high amounts of side effects. So between all these guys that we've pointed at so far, the cell membrane, cell wall, ribosomes, which one of these is going to have the highest amount of side effects? Yes, the cell membrane. Exactly. Perfect. Good. Good, good, good. Now, viruses. Completely different than the other two. Viruses are set up, first of all, they don't reproduce by themselves. They're very small compared to your regular bacteria, as well as your regular eukaryotic cell. And they hijack their mechanisms. Okay? For prokaryotic, they're called prophages. Okay? Now, for eukaryotes, there's no specific name for them. They're just viruses that you have to know. But for prokaryotes, there's a totally different category, which is known as prophages, because they only target prokaryotes. They only target bacteria. Okay? Good. Any questions on any of this so far? Okay, good. Now, yes, prophage is the same thing as a bacteriophage. Yep. Same thing. Good, good, good. Okay, now, here's this lovely category. And I'm going to tell you, this lovely categories is going to be your Bible when it comes to bacteria. Okay? When it comes to viruses, yeah, it's a still kind of concept, but it's a little harder with viruses because there's so many viruses in each category. In some of those categories, I say yes. But here at bacteriology, trust me on this, this is your Bible with it. You must know this categorization. If you do not, it will hurt you dearly. Okay? Because this is how they're going to test you. They're going to tell you that this bacteria that they've colonized is gram-negative in rods. And then they're going to say, what bacteria is it? If you don't know, generally speaking, what bacteria are gram-negative in rods, you're going to get screwed here. Okay? So that's why I'm saying you have to have this down pat. Now, with gram positives, I'm not going to go through this whole list because it's just reading off a page. But the way this is broken down is broken down into rods, which are known as bacilli. Why is my word so big? Rods, which are known as bacilli, cocci, which are known as circles or spheres. Okay. And then you got branching filaments, which are rods that are linked together to form branches. Okay. Now, the other categories that you have things like diplococci. These are two spheres connected to one another. Okay. Then you have coccobacilli, which are a fusion or a mixture of sphere and rods mixed together to the point that they don't really look like rods. They look more rounded, oval shaped. So they gave it a cocci or coccobacilli nomenclature. Okay. Yeah, they're like pseudo rods. Perfect. They're like rods, but they're more spherical, like. Okay. But that's what coccobacilli are. All right. Now, with that being said, you can see coccobacilli isn't broken down any further. It's just what they are. Now, the next group that you'll need to know is curved rods. These are rods that instead of being directly straight like this, they kind of look more like this. Okay? That's what this category is broken down into. Okay? Now, with that being said, this is going off the nomenclature that we've stained this with what is known as a gram stain. Things that do not gram stain are not in this category. And there are bacteria that do not gram stain. Okay? For instance, your Mycobacteria tuberculosis, they do not gram stain, which is why you do not see that anywhere in this category. Okay? That doesn't mean that they don't exist. They do. They just don't gram stain. Okay? Which is why they're not on this list. Okay? They stain a different way, which is known as what? Zeo-Nielsen. Also known as acid fast. You do need to know both names. Okay? You need to know both names because that's very important. Because they could say Zeo-Nielsen stain, they could say acid fast. But yes, that's how that's set up. Now, let's move on. So the way I'm going to advise you to do it is this. When you're going through your semester and when you're going studying through your class, like now prepping, don't sit here and be like, I'm going to do all bacilli. Let me do all that. Listeria bacillus conibacterium. Cool. Clostridium cutibacterium, which was formerly known as Propionibacterium. Cool stuff. Now, what I advise you to do is this. I advise you to do your specific sections as you go. So for instance, your first test, you won't have any of this really. You're going to have gram staining. You're not going to have any bugs to remember, but you will have gram staining and you will have the basics of bacteriology, which I'm going to go over. The second, third, fourth test, you'll have bacteria and viruses. When you go over them, look at this table, and I want you to follow down with the table to their respective guys. So you're like, oh, this bacteria that I'm learning about, let's say for GI, right? You'll learn about, or your next block 2, you'll learn about staph. So you'd be like, okay, so it's a cocci that's catalase positive. Oh, all staph are catalase positive. Okay, that's cool. And then you'll be like, oh, well, this guy's the only bug that's coagulates positive. That's very important. Whenever you see something that only one bug is noted inside of a certain test, they can just essentially give you that the bug is coagulase positive. And you should know right off the bat what that bug is. Or they could say something like, the bug is novabiasin sensitive. Right away, you should know what bug that is. Okay? So whenever you have one bug in a category, make extra effort to memorize that bug with that test. Okay? For instance, if you see this, they can't use that as a way because there's too many bugs in that one test. All right? Good. That's the other thing I really want to stress with you guys. All right, then. Let's move on. Ooh. Okay. So now let's talk about the different aspects. So the structure of bacteria. So bacteria, they have different structures to them. And on top of that, they have something that's very unique to them. Okay, this is bacteria. This is not viruses, not fungi. Okay, specifically bacteria have a unique characteristic that they become what is known as a spore. Okay, now, not all bacteria do this. Only certain bacteria become a spore. Is that important to know what bacteria become a spore? Heck yes. You do have to know that. Because these spore guys are the guys you're trying to kill when you do something known as autoclaving. When you autoclave something, you put something at a very high heat for a very long period of time. Yeah, you're essentially ovening them, but at like very high heat oven. Like we're talking like those people who make glass type ovens. And they're like melting the glass and like forming pots and stuff like that. Like you're getting very high heat for a very long period of time. Okay, that is what is known as autoclaving. All right. Now, there's something in it that makes them very resistant. This is what is known as dipicolinic acid. This protein is what is responsible for making these spores extremely resistant. Okay? And you can see how this is only in gram-positive bacteria, particularly the Clostridium family. And you guys will know more about that as you study. Okay? But take very key, pay attention whenever someone says this is a spore, or this comes from a spore, or a spore forming family. Pay very particular attention to those, because this is why we do autoclaving in the medical field or in research, period. Okay? Now, some bacteria, they also have the ability to move about, which is these flagella or flagellum. Now, some bacteria may have many, some bacteria may have one. Do you have to remember that? Not really. You just have to remember what bacteria are motile. That is something that's very important. Okay? With that being said, you'll run across something like this and hit a Hectoran auger, or also known as a Hectoran enteric auger, which is used to study for motility. Okay. But yes, let's keep moving. You'll run across this later when you get to that portion with GI. Now, pilus of fimbriae. These guys are very important for adhering to the surface of cells. What do you think, like what type of cells do you think would need? Like what type of infections? Do you think skin infections would need something like this? Not really. Where are you constantly like moving stuff around? You're constantly pushing things down. Yeah, your stomach. Perfect. Think of wherever you have things constantly moving across it. The skin, not so much. Yeah, you got your skin sloughing off and so on and so forth. But even if you had pili and fimbriae, all that would do is latch onto that skin cell that was falling off anyways. That's not really going to help you, right? Like that doesn't cause an infection. Okay, but places like GI system, where fluid is constantly being flushed out of the GI system, You having the ability to latch onto the walls is very important. Okay. Same thing with kidneys and UTIs. Perfect. Good. So this is where you get a lot of the GI infections, a lot of the urinary infections. Where else do you think is very important? Other than GI and urinary. Where else? Respiratory. Perfect. These are where these guys are very important. Okay. For latching on and holding on. Okay. Good. Now, let's keep moving on. Okay. So this is breaking this down even more to the different parts of the bacteria, right? We talked about the fimbriae or the pili. We've talked about the spores. This is a picture of a spore-forming bacteria. Are they ever going to show you that picture? I've never seen this picture. Is it possible? Yes, it's possible to still see it because this is coming from first aid, but it's not something that you're guaranteed to see. But you can see these guys here, Clostridium family, very important. The other one is the Bacillus family. That's the other big guy. Those are the two families that are spore-forming guys. Clostridium, and the last one being the Bacillus family. Bacillus anthrax, Bacillus cereus. Okay, good. Now, let's keep going. The next thing that's also part of this bacteria, outside of, so here's my little bacteria, right? We have these pili, allowing it to attach to the walls. It can become a spore and then come back to a bacteria when the environment is more favorable. The other thing is that it can also have around it a polysaccharide capsule. And this polysaccharide capsule is almost like slime. Okay? It produces this extra layer around it that prevents phagocytosis from happening. That's essentially what ends up happening. Okay? So that's what this polysaccharide capsule is. This is where antibodies come into play, right? That's very important because it binds to these polysaccharide capsules and then allows these macrophages to eat up these microbes. What's the CD guy that these FC portion binds to? CD 16. Perfect. Good stuff. CD16. So it binds to these CD16s and then they eat them up. Okay? Via that antibody dependent cellular cytotoxicity, ADCC. Now, let's keep going. The next thing is this thing called a protein A. This is very unique to only one bacteria. So, yes, it is important. I'm not saying it's not important, but it's only important for one bacteria. For the grand scheme of things, all bacteria do not have this. Okay? But essentially, all protein A is that it allows it to prevent phagocytosis, just like the polysaccharide capsule. I'm not going to concentrate so much on this because this is very unique to only Staph aureus. You will learn this more in depth when you go over this in your second test, CAS 2. Okay? You'll learn more on how protein A works and what is its purpose. But in the grand scheme of things for your first test, they're going to ask you things like, what is a capsule? What's a polysaccharide supposed to do? What is that preventing? How do you combat a bacteria that has a polysaccharide capsule? Antibodies. B cells are very important. Plasma cells are very important. To be even more specific, is it antibody dependent or antibody independent? Oh, not antibody. T cell dependent or T cell independent? More important when targeting polysaccharides. Which one's more important? T cell dependent or T cell independent activation of B cells? Why? Why is T cell dependent more important than T cell independent? Well, IgM, if you remember, IgM is the big guy that activates complement. And if you remember, IgM and complement is only really big when it comes to gram-negative bacteria, not gram-positive bacteria, right? The only thing that really helps you with polysaccharides and IgM is you get C3b. Because if you remember, C3b is an opsonin. And that's the only thing that's really going to help you with a polysaccharide capsule. Yes, perfect. The MAC isn't really going to help you that much unless it's gram-negative. If it's gram-negative, then I say yes. But if this is a gram-positive bacteria, MAC isn't really going to help you. Now, the next thing is antibody dependent because IgG. If you remember, IgG is the big guy that goes through antibody-dependent cellular cytotoxicity. He's the big guy that activates the FC receptor with CD16 and does all that stuff. So that's why T-cell dependent pathway is very important for breaking down bacteria that have polysaccharide capsules. Okay? All right, good. Now, the next thing I'm going to talk about is IgA protease. If you remember, IgA is something that is released into the mucous membranes. Right? Now, I didn't go in depth on how IgA gets there, but if you remember, IgA is a dimer, right? You have one IgA here, and you have another IgA here, and then they're interconnected by this thing, known as a J chain, right? This is IgA. Now, IgA protease, what essentially IgA protease does is it damages the J chain and destroys it. So now these dimers become monomers. Now, dimers becoming monomers, why would that be an issue? This is an issue because the J chain is responsible for not only causing it to be a dimer, but it also prevents it from being degraded by all the other innate immune system inside your stomach lining. Okay? As well as allows this IgA to anchor itself on the epithelial cells. So if you lose this J chain, this guy now gets washed away. All right? That's essentially what happens. You wash it away in your feces, you wash it away in your urine, you wash it away in your nasal drainage, You wash it away, when you cough up mucus, whatever. Okay, you're losing it. It's not staying where it needs to be. Now, this IgA protease are mainly noted in bacteria that colonize those areas. Why am I going to have an IgA protease if I'm not on mucous membranes? If I'm targeting your skin, there's no reason for me to have IgA protease because there's no IgA there. Okay? So as long as you have an understanding of where you're at, you will always know if this is IgA protease positive or negative. Okay? So, for instance, things like Streptococcus pneumoniae, that's a respiratory thing. Haemophilus influenza type B, also a respiratory thing. Neisseria also targets the respiratory system. So they all have IgA protease. Okay? Good. Now, M protein, this is very important. So in the urinary system, IgA is there, but in the urinary system, when you get to that portion with the microbes, they don't really have that much IgA protease. Instead, they do it a different way. They have fimbriae and pili, and these fimbriae and pili, they change. They're very mutagenic. If you remember, antibodies target one specific antigen, right? Now, if you mutate that antigen, that antibody essentially becomes obsolete. And that's how these bacteria in the urinary system work. They don't bother so much with IgA protease. They bother more with mutating these fimbriae and pili. And that's how they constantly avoid the immune system. Okay? And the reason why that is because you got to think, urine is very acidic. It has a lot of other things in it as well. And on top of that, you also have bacteria lining your urinary system. Okay? And they are just chilling there. Naturally occurring bacteria that don't really do anything. They don't cause damage to your body. Right? So if you had IgA protease in those locations, you're going to have a lot of issues with those normally occurring bacteria staying there. Okay? But essentially, that's what's going on. All right, perfect. Now, M protein, I'm not really going to harp on too hard. You will know more about this when you do your second test, CAS 2. You're going to talk about M protein. You're going to talk about Streptococcus, pyogenes. But this isn't a whole bunch of bacteria, so I'm not going to harp on it. You're not going to see this on your first test. Okay? You'll see this more on your test coming onwards. Now, the next thing that's very important to know is this concept of endo versus exotoxins. This is something you will get tested on. Endotoxins are toxins that are produced by the bacteria, but is a part of the bacteria. For instance, if this is a bacteria and I produce my cell wall, this is an endotoxin because I'm producing something that is essentially part of the bacteria. Okay? It's a part of the bacteria. Now, exotoxins are not part of the bacteria. They are something that is released from the bacteria that leave the bacteria to do other things. Okay? So what I want you to kind of think of this in synonymous with what you already know from year one, think of these guys as cell signalers. They are got cell signals or cyclic AMP, stuff like that. It's all inside, right? But when you look at paracrine hormones, they leave the cell and they go do their function. Okay? That's exotoxin versus endotoxin. Now, my question to you is this now. If a cell is sitting there and you have a fever due to the cell, is this because of an exotoxin or an endotoxin? You are not killing these bacteria. These bacteria are just sitting there and you have a fever. Is that due to an exotoxin or an endotoxin? Exotoxin. Perfect. When do you start seeing things pertaining to endotoxins? Yes, so these exotoxins do cause metabolic changes. They do cause issues in the body. For instance, I can give you an example. Tetanus toxin. That's an exotoxin. Botulinum toxin. That's an exotoxin. Okay? So these are toxins that they produce, right? And is not a part of them. Now, endotoxins are only released when these guys die. When they die, this cell fragments into little parts. And these little parts are endotoxins. Okay? Does that make sense? Okay, good. Now, these endotoxins are very dangerous. Why? Because they cause massive immune response. They cause massive immune response. Yes, like LOS, lipooligosaccharides, or LPS, which is also one of them. You even have what is known as a lipid A, which is also an endotoxin. These guys are released whenever the cell wall breaks down because the cell bacteria dies. If you never kill the bacteria, you never have a massive immune response. Okay? Now, with that being said, there's a lot of research going on. I'm just going to say this one little bit. There's a lot of research going on with people who are immunodeficient. If you're immunodeficient, you're not really essentially killing the bacteria. So if you never kill the bacteria, that's why people who are immunodeficient, sometimes they're sick and have illnesses, but never really have a true fever. Because they're not killing the bacteria, releasing these massive immune response, causing a fever. Okay? And that's the new stuff going on with the research behind that. But it's based on this concept. So make sure you don't forget it. Now, this massive immune response can lead to what is known as septic shock. Okay? Essentially, what ends up happening is you have massive vasodilation. You'll learn about this more in year 4. I'm just talking about it now. Massive vasodilation that tanks your blood pressure. And it's because of this. Lipid A, lipooligosaccharides, LPS, endotoxins. Okay? All right, good. Now, we've talked about all that stuff. Now let's keep moving. Now, here we're looking more at the envelope or the cell wall aspect of things. I'm going to finish off this and then we'll take like a 5-minute break. You're looking at the cell wall portions of the bacteria. Okay? There's different parts to them. We already talked about how they can be capsulated with a polysaccharide capsule. I'm not going to talk more about it. We've already harped on it. Now, the other thing they can form is what is known as a glycocalyx. Now, this glycocalyx isn't something new. Your normal cells produce this as well. They produce this thing known as a glycocalyx. Essentially, all it is just an outer layer to your cells that are formed up of sugars. Now, what this essentially does is it causes you to have this area around the cells which are water soluble. Okay? And that's essentially what ends up happening. And then if you remember back in year 1, when you talked about GI digestion, that was why you had issues absorbing fats unless you had bile. Because bile makes it water soluble. Now it's water soluble. So now we can actually transverse the cells, passing this water soluble zone from this glycocalyx and getting inside the cell to be formed into triglycerides and pumped into the lymphatics. Okay? So this isn't something new, right? Now, let's keep going. The next thing is this concept of an outer membrane. Not all bacteria have an outer membrane. What bacteria have outer membranes? Gram-negative. Perfect. Yes. Gram-negative bacteria have an outer membrane. Gram-positives do not. Good. This is why gram-positive stain positive and gram-negative stain negative. Because they have an outer membrane versus the gram-positives that do not. Because essentially what you're doing when you're doing a gram stain is you're staining this thing here, which is known as peptidoglycan. You stain the peptidoglycan, and it makes it turn up blue or purple, whichever one you want to go with. Versus with gram-negative, you have this outer membrane to it. Therefore, whenever your gram stain tries to come, it gets blocked off. And it can't get to the peptidoglycan inside, so these never get stained. Okay? So that's why gram-positives are positive and gram-negatives are negative. Now, let's continue. With you having this outer membrane, you produce also what is known as a periplasm space, because now you essentially have two plasma membranes sandwiched on top of each other, which is space in between. Now, this space in between is very unique because what can happen is this. Bacteria are always wanting to protect themselves. Just like your regular cells are always wanting to protect themselves unless they get virually infected and they can't help it, right? And then they tell other cells, hey, I'm infected via interferons. But outside of that, they always want to help themselves. So what ends up happening is bacteria release enzymes that break down drugs. Okay, they degrade the drugs that you're trying to use to kill them. However, here's the issue. If you have a cell here, a bacteria, that does not have an outer membrane, and it releases an enzyme, what's going to happen? It's going to diffuse across high concentration to low concentration. So as a result, technically the bacteria doesn't have any enzyme around it, even though it's trying to protect itself. However, in a gram-negative bacteria, you have this periplasm space now because you have the outer membrane. So now what ends up happening is it secretes an enzyme and it gets stuck in this periplasm. This is why gram-negatives are so resistant to antibiotics. Okay? Good. Any questions on that before we move on? All right, perfect. Now, we've talked about the periplasm now. We've talked about the cell wall being that outer portion. And now the last thing is what is known as the cytoplasmic membrane. Now, the cytoplasmic membrane is the actual membrane of the bacteria. What's unique about the cytoplasmic membrane for bacteria? What's unique about it? That makes it different than the cytoplasmic membrane of eukaryotes. How is it different? Or are both cytoplasmic membranes the same? So the big difference between the two is with bacteria, the cytoplasmic membrane has the electron transport chain in it. Versus our normal cells do not. This is why there's that whole theory on how a bacteria got trapped into one of our normal cells and became what we know as the mitochondria. That's why there's that theory behind that. And how that then was what helped with, the whole evolution and how that whole thing worked. But that's what that's all about. But essentially, bacteria, the electron transport chain's on the cytoplasmic membrane. In eukaryotes, it's not. Okay? That's the big difference between the two of them. All right? Now, let's continue. Now, we've already talked about the capsule. We've talked about the outer membrane, vaccines, we'll talk about here in a minute. Okay, good. Let's take our 5-minute break. When we come back, I'm going to talk about the little small details pertaining to each one of those guys, now that you have a good idea of everything. And then we'll be pretty much done with bacteria, and then we'll start moving into viruses. And then fungi. And then we'll be done, pretty much. All right, good. Five minutes. See you guys in a bit. If you have any questions, you can ask me now. All right. Let's finish this off. Okay, so these are just the remaining little details pertaining to each one of these guys that are also very important to know, that you don't just kind of gloss over them and move on, but you will also kind of harp on this as you go the whole year, actually, for Med 3 and for Med 4. They're going to talk about this. Okay. So with the capsule, the one thing that this thing can do is it can also form what is known as a biofilm. Okay. Now, this biofilm is essentially just multiple bacteria that have formed an outer covering of polysaccharide. This is no different than the plaque you see on your teeth in the morning. Okay. Or at night, whichever one, right? This is essentially what that is. It's bacteria that have secreted their capsules and have formulated this outer kind of slime layer that prevents it from being phagocytosed. Okay. Now, with that being said, you can see how this can be problematic, because if antibodies try to bind to it, it's way too big for a macrophage to eat it. And it's not like it's just going to take a little small chunk out of it, and then a little macrophage is going to come over again and then take a little small chunk out of it, and then eventually they're going to eat it away. No, it doesn't work that way. Okay. Macrophages essentially eat the whole cell. Okay. That's foreign. Now, with that being said, this comes into play when you look at things like catheters, when you look at things like heart valve replacements. Because essentially what ends up happening is this: these things end up getting biofilms on them. Now, all of a sudden, it's hard for you to autoclave them. It's harder for you to remove these bacteria from the surface of these tools, medical tools. So whenever you place them inside of the human of your patient, right, after surgery or during surgery, now all of a sudden, they are infested with this bacteria. Okay. This is why they get infected after prolonged usage of catheters, or after being having a heart valve replacement. Whenever you see someone who comes in, they just recently had a surgery, now they have a fever, you got to think that it's one of these bacteria that forms a biofilm. That's why he got this infection. Okay. All right, good. Now, the next thing we've already talked about is vaccines. Okay. And we've already harped on the difference between conjugated vaccine versus unconjugated vaccine. What's the big difference between conjugated versus unconjugated? Yeah, you have some kind of protein with it. Perfect. Conjugated vaccines, they are sugars plus a protein. With unconjugated vaccines, these are just proteins, right? You use these conjugated vaccines because sugars are things that do T-independent activation. So as a result, you have to add the protein to change the T-independent into T-dependent pathway, right? Good. Now, cell wall, we've already harped on. The only thing added on, I'm going to say, is this thing known as lipoteichoic acid. Lipoteichoic acid is also something on the cell wall outside of the peptidoglycan that activates macrophages. Okay? And then you already remember LPS, lipopolysaccharides. This is the guy that deals with CD14 on macrophages, right? They get activated, and then they send the signal inwards to NF-kappa beta. And as a result, you formulate all those lovely cytokines and interleukins. Okay? Good. Now, let's keep moving. Here is how the lipopolysaccharide, or LPS, is formulated. Okay? You have what is known as this thing, lipid A. And if you remember, this is a endo or exotoxin. Endotoxin, perfect. This is an endotoxin, and this endotoxin is responsible for causing all of that immune response and activity. Okay? Only happens when these cells die. Okay? Now, you also have something on here known as the O antigen, which is where antibodies can bind to on these lipopolysaccharides. Okay? Now, here, nothing really different here. This is a throwback to the immunodeficiencies that we talked about. If you have T cell deficiencies, what type of infections do you get? Viral and fungal. Viral and fungal. Very important with T cell. Okay? If it's B cell related, what is it? Bacteria and parasites. Good. Good, good, good. This is the biggie, right? On how you differentiate. Is this a T cell related? Is this B cell related? Is this both? Right? And if it's innate immunity, then you only take one of them. For instance, it could be just viral aspect, or it could just be fungal aspect with neutropenia, or it could just be a bacterial thing with things like Chetek-Higashi syndrome. Okay? So that's how that breaks down. Make sure you don't forget that. Okay? Now, staining, we're going to talk about it. We've already talked about gram stain, gram positive versus gram negative. Okay? And there are some of these guys that do not gram stain at all. There's a mnemonic for it. You can use it to memorize it. It's up to you. I say you work through it, as you go through the classes. And whenever they say something does not gram stain, you just take a particular emphasis at that point in time. And then you add it and you make the list that way. Because if you try to just throw them all in now, it's hard for you to remember these guys. because you don't understand what they do, where they're coming from, or anything. You just know that they don't gram stain well. Cool. That doesn't help you out on a test. Okay. Now, Giemsa stain is very important for bacteria or parasites that are intracellular. Okay, these are the guys that are noted inside of cells. Okay, because in gram stain, you never find them because they're protected by your own cell's membrane. But in Giemsa stain, you stain them that way. Okay, PAS, we already talked about, and you know that with glycogen. Zeal-Nielsen, also known as acid fast. Very important. Also known as carbofusion, also important to know. Silver stain. This guy here is really good for very small organisms. Now, with that being said, you're probably saying, well, all these organisms are small because I can't see them with the naked eye. So, yes, but there's some of them that you can't even see with a microscope. That is what we mean by small, as being very small. If you smear it on a microscope and you still can't see it, that is very small. Okay? That's the difference. Now, with that being said, yes, fungi are in this category, as well as two bacteria, Legionella, H. pylori, as well as the spirochetes. They also fall into the same category. Things like Borrelia burgdorferi, the Lyme disease, things like leptospirosis, also something like along the lines of syphilis, also silver staining. So we'll use silver staining to check for ulcers. I don't understand your question on that one. What do you mean by ulcers? Because ulcers is something that you can visibly see. Oh, so you'd only do the silver stain if it was something that you can't see with a light microscope. Essentially, when you are looking at an ulcer, a lot of times you don't have to do silver staining unless, yes, it's in the stomach with H. pylori, because H. pylori is something that does produce ulcers in the stomach as well in the intestines, but is not visible to the eye. So in that case, yes, but just normal ulcers on the skin, normal ulcers in your mouth, no. But ulcers in the stomach, yes, I do agree. You would do a silver stain. Yes, yes, yes. Good. Okay, so let's keep going. Now, the last thing is this thing known as fluorescent antibody stain. This is a little different. Essentially, what's going on here is you take this microbe that you're looking for, okay? And you take an antibody that binds to this guy, okay? And what you do is you take the stain or whatever culture, whatever sample you're using, and you add this antibody to it. So now you have this bug with this antibody attached to it. Then you tag it with a fluorescent dye or an enzyme that whenever you get a certain substrate in there, it produces a fluorescence. But that's essentially what ends up happening. So with that being said, you can see how this can be used for a wide range of things, not just one type of bacteria, because you're essentially seeing if the antibody binds to the bacteria. If the bacteria is there, it's there. If the bacteria is not there, it won't bind, right? So that's why it's used to identify many bacteria, as well as viruses, as well as parasites. And that's why, because essentially you're just using antibodies to bind to whatever you're looking at. Okay? Good. Now, with that being said, if you see that there's a positive fluorescent antibody study, does this mean you have a high sensitivity test or a high specificity? This is a high specific test. Okay, because it's very specific for this bacteria, very specific for this virus, very specific for this parasite. Okay, specificity, well, this is kind of throwing you back to biostats, rules in disease. So when you go through studying, make sure you do not gloss over these. Okay? If there is a test that's very specific for a diagnosis, jot it down. Okay? Make the effort to really drill that specific test in your brain. Because if they ever say that anywhere in any question, then you always know what the diagnosis is. Because that test can only be one disease. Okay? So that's why I'm saying jot down the specific test because they're very important. Okay? All right, good. Now, here's this last bit with bacteria and the growing aspect of it. Bacteria, they divide, right? One cell becomes two cells become four cells become 8 cells. It's exponential, right? So essentially, that's what's ending up here, is you originally start off with just kind of flatlined. Okay? Nothing's really going on here because the bacteria are just making the product like proteins ready to be utilized for division. Then all of a sudden, they get all the proteins they need, and then they start to divide, which is known as the exponential stage. And then they get to a point where they've used up all the nutrients. And now, for them to continue growing, some of them have to die, which is known as the stationary phase, which is why at this point, growth is equal to death. One cell has to die for another cell to grow. Okay? Now, eventually what ends up happening is even those nutrients wear off and wear down, that when the cells die, there's still not enough nutrients. Okay? And as a result, you're in a death phase. You're going downwards. Okay? Now, that's the process. Okay? Now, another way you can see it is something along this. They could take this word out here, and then they could ask you, A stands for what? Or they could say, B stands for what? Or they could say the things like antibiotics would be good for what stage, A or B? Okay? So essentially the way you see that, this number here is the same. So that means that this here is known as the stationary phase. Well, not stationary, the lag phase. It could be stationary as well because it's the same, right? You're dying and you're forming, but it's the lag phase because afterwards you have growth. okay? Which is known as the exponential phase. So in the lag, yes, there's no replication going on. You're just preparing to replicate. Exactly. Now, do you see symptoms at this point in time? No. Perfect. So I want you to connect this lag phase with also the lag phase you get between getting exposed and showing symptoms. Okay? This is what is known as the lag phase. And no, not necessarily. It's not equivalent. And the reason why that is because the number of bacteria cells needed for you to eventually show symptoms may not be the same number as what you got inoculated with at the beginning, right? You could have got inoculated with only 10, but then you need 10,000 or 10 million to show symptoms. Okay? So not necessarily the same. But yeah, good. Good. Now, let's keep moving. And yes, you are right. Antibiotics are really good during the doubling time. Okay? Now, there's different types of bacteria, obligate aerobes, facultative anaerobes, intracellular bacteria, we already said. This is more, once again, just an identifier for it. This is inside this lovely thing. And you can see that, and you see how important this list is now. Aerobic, anaerobic. Okay, so that's why it's very important to know this list, because all that stuff is literally inside that list. Now, I'm going to touch on this real quick because this is important when it comes to antibiotic resistance. So, whenever a bacteria cell, here's one bacteria cell, bacteria A. Here's another bacteria cell, bacteria B. Okay? Let's say this bacteria A dies. He dies. And as a result, he releases his DNA when he dies, right? DNA comes out of the cell. Nothing new here, right? Cell B can decide, hey, let me take in this DNA. And now bacteria B has B DNA plus A DNA. Does that make sense? Okay, good. Now, there's one other way this can happen. Let's say bacteria A, bacteria B. Let's say bacteria A instead gets infected by a bacteriophage or a prophage. Okay? Now, as a result, cell A ends up dying, releasing a whole bunch of viruses. And one of the viruses can accidentally be infused with A's DNA. Does that make sense so far? During the packaging of these viruses, sometimes bacteria DNA can get trapped inside of a viral capsid. Okay? To the point now that this guy, when infecting B, now becomes a bacteria with A DNA plus B DNA. Okay? So this is the second way this can happen. Now, the last way you can end up getting A and B DNA together in one bacteria is this. A DNA, B cell. What can end up happening is if they are the same species. Very important. Same species, they can end up communicating with each other via, yes, what we know as pili. Perfect. Now, as a result, some of A's DNA goes to B's DNA cell, and you get a bacteria with A plus B. Do all three of those make sense? Okay, now, this is same species. These other two do not have to be the same species. Okay? Essentially here, all you're doing is taking up DNA in the environment. Here, you're getting infected with a bacteria, or no, a bacteriophage or prophage, which is causing you to then have the DNA. You do not have to be the same species of bacteria. Okay? This is how antibody resistance is passed on from one species to the next. Okay? Now, let me talk about actual names to make sense of this. The one that you're picking up DNA from the environment, that is known as transformation. Okay? Now, the one where you have some pili that connects the two together, that is known as conjugation. Okay? Now, the last one that we want to talk about is what is known as transduction. That is the one where you have the virus infecting one, getting some of the DNA, going to another bacteria, and pushing that DNA into the new bacteria. Okay, so now the new bacteria has A and B. All right, those are the three ways: transformation, conjugation, transduction. Any questions on that? Okay. Good. Now, we've already talked about antibiotic resistance. That's how it does it. It's transferring over in that DNA the nucleic acid arrangement for an enzyme. And that is what is then formulated in the new bacteria as the enzyme, which then produces a resistance. Now, let's talk a little bit more. What he said about transduction and stuff like that. Let's talk a little bit more about bacteria being infected with viruses. Now, the two ways that bacteria and viruses can interact is one known by lytic. The lytic is the one where they explode. Okay, they get infected with a virus. The virus replicates itself, and whenever it's done, it just lyses the cell, the bacteria, or our human cells, and allows that virus to then be released. Now, the second way a virus can infect you is it infects you, and instead of formulating new viruses, it decides, hey, let me instead go into the DNA and let me chill in the DNA. So now my question is this. If you get cell A, cell B, here's our DNA. And here's our virus. Okay, this virus goes in and then becomes part of the DNA. This virus comes in and then becomes more virus to rupture the cell. Which one of these has a higher risk of becoming cancer? Do not forget this. This is why any viruses that are DNA-based get incorporated into the DNA. They then activate cell cycle regulators to allow the virus to be duplicated as well, ends up increasing the risk for cancer. For example, HPV, cervical cancer, it's a DNA virus, does this exact same thing. HIV, same thing, goes into the DNA, causes it to be fused with the DNA, and then activates its own cycle to increase risk for cancer. Last one, EBV, same concept. It goes, it's a DNA virus, goes inside the genome, intercalates and adds itself to the DNA of your normal cells, and then causes that issue with cancers. This is why this happens. If you do not incorporate that virus into the DNA, it will not have a higher risk for cancer. So that means RNA viruses, that means single-stranded RNA viruses, which I'll talk about later, do not have any risk for cancer. Only DNA viruses do, because they're the guys that go into the DNA, right? Now, HIV is a little different because it's an RNA virus that becomes a DNA virus via reverse transcriptase, but it's the same concept. If it's a DNA virus in some portion of its cycle, it can incorporate into our own DNA. Now, that's that concept. Good. Very important to know. That's that tie-in to neoplasias that we'll talk on more when we get to the end. Now, antibiotics. Okay? With antibiotics, I'm sorry to say, a lot of this is just memorization. Okay, you got to know the antibiotics. You got to know how they work, the mechanism of action. There's no way to get around it. Okay? There's different types, but essentially you got to think of it in this manner. We want to target things that are different than our normal cells. That's what we do. We target things that are different than our normal cells. For instance, the cell wall. For instance, the cell membrane, right? It has that electron transport chain there. Our normal cells do not. For instance, this thing here with folic acid, PABA to DHF or dihydrofolate is an enzyme that our normal cells do not possess. Okay? And you'll see that when you get to that point. Okay? Now, here, trimethoprim, this is an enzyme that we have, dihydrofolate reductase. Okay? So, there's a lot of side effects with it. But we never use it by itself. We always use it with sulfamethoxazole. Yeah, disulfiram is the name of it. Now, let's continue. So with that being said, we're just going to keep going. I'm going to harp on the major side effects of each one of these guys as we move on. But I'm not going to spend too much time because it literally is, you just got to study it and you got to remember it. Okay. There's not any other way around it. Now, here you got the protein synthesis inhibitors. What are they targeting? Ribosomes. So what do you think is going to be the main outcome of this? Decreased protein synthesis. If you're decreasing protein synthesis, does this kill the cell? Or does this just make the cell not produce proteins? Yes, good. No death. Perfect. These are the details you need to make sure you remember, because some of these things kill bacteria. Some of these things do not kill bacteria. They just stop them from dividing. Okay? If you are someone who's immunocompetent, so that means someone who has a competent immune system. You want to not necessarily kill the bacteria. You can just stop them from dividing, and your immune system will just do what it does. Okay? But if you are immunodeficient, then you want to kill the bacteria, because his own normal immune system won't kill it. You can stop it from dividing, which is all good and well, but that they're just going to still chill in him, and they're still going to do whatever they do. He's still going to be infected. Okay? All right, good. Now, let's keep moving. Good. Now, let me think here. I'm trying to see if there's anything else I want to say about this. No, because a lot of this stuff you're going to end up seeing as you go. Like, for instance, penicillins, they have high risk for having hypersensitivity reactions, as well as allergic reactions. Penicillins. You have to know, for instance, where is it? Vancomycin is really big with what is known as red man syndrome. Let's see here. Aminoglycosides are very big and toxic to your kidneys. So they cause kidney damage. Really big kidney damage, guy. Macrolides lead to GI and cardiac issues. Fluoroquinolones are very big with causing tendon rupture. Yes. Metronidazole is very big with a disulfiram reaction. What that means is people who drink alcohol get very sick. Okay. So will they ask questions like fluoroquinolones, like tendon rupture? Yes. So what they could ask is this type of question, and you'll see these questions when we go over it. They could give you a vignette where the patient is taking a fluoroquinolone, and then all of a sudden they get ankle pain, and they're asking you what is the most likely cause of this guy's symptoms. Or they can say this patient's taking all these drugs and he gets sick. They give him a prescription of another drug, and then what is the guy that he was most likely prescribed with that caused this issue, right? So those are the type of things that they can ask pertaining to side effects. Okay. Bacteria, viruses. A lot of stuff we already hopped on and we talked about. Here with the viruses, there's a lot of different categories for viruses. The viruses can be DNA. Viruses can be RNA. And if you remember, DNA and RNA can be single-stranded or double-stranded. So can be single-stranded or double-stranded. Can be single-stranded or double-stranded. That's all your categories, right? So we have single-stranded, we have double-stranded DNA, Then we have double-stranded RNA, single-stranded RNA. But then we end up seeing this plus and minus. What the heck is that? So the difference is that RNA, if you remember, when it becomes mRNA, it's known as a template strand. And this template strand can become protein. Now, you also have RNA that can be complementary to this. However, when you are complementary to this mRNA, AUG becomes UAC. This is no longer a start codon. Does that make sense? Now, that means you do not make protein. So this is what is known as the negative sense RNA. Now, the next thing is negative sense RNA. When you infect it into a cell, will you end up producing virus? If you take a negative sense RNA, no, perfect. But if you take a positive sense RNA, you will produce virus. So how do these negative sense RNAs do their job? The way they do it is the negative sense RNA carries with it an RNA dependent RNA polymerase, which takes this and changes it into a positive single-stranded RNA. And this then causes infection. Okay? Any questions on that? Now, does that mean that this is only present in negative sense RNAs? No, perfect. Because also in positive sense RNA, you need to replicate the positive sense RNAs. So it does that by using this template to form a negative sense RNA, which it then utilizes to form a positive sense RNA, just like negative sense RNA form positive, which causes producing of proteins, and then uses the RNA-dependent RNA polymerase again to become the actual virus genome. Okay? So these guys are your actual virus genomes. So that's why you use RNA-dependent RNA polymerase twice. Once, either to form the active RNA that can form protein, and then form the genome of the virus, or once to become the template to form the genome of the virus, because this here can form the protein by itself. All right, good. Now, with that being said, a virus can have an envelope or cannot have an envelope. Which one do you think is more sturdy? It seems a little redundant for the positive. The only reason why it has to do that for the positive sense is because there's no way for the positive sense to replicate itself. There's no way for it to replicate itself. So it has to form the negative sense to then replicate off that. There's no positive sense to positive sense because remember, everything's complementary when you're thinking of DNA, RNA, like replication stuff. All right. Okay, good. So which one do you think is stronger? Something that has an outer membrane to it that is liquid phospholipid fats, or something that is strictly just protein outer layer? Okay, so now this is the big thing I need to ask you. If you have an outer envelope that's lipid and fluid and needs liquids, it needs to be wet for it to work. Do you think this is very, do you think if I was to touch my hand on the table, that's a dry table, lift my hand up and look at it under a microscope, would I have positive enveloped viruses or naked viruses? Naked viruses. Perfect. The reason why is because all those enveloped viruses are gone. They can only survive whenever they are in a liquid medium. Okay? Only when they're in a liquid medium. So that means they're not very, it's harder for you to catch these guys. The only way you're going to catch them is if you're going to respiratory droplet. Like he has to actually get it via that way. Yes, exactly. COVID falls into this category. Perfect. Now, naked viruses are different. They are guys that are very strong because they don't need an outer layer of liquid. So that means they're very resilient. And these are the guys that cause GI symptoms because they can withstand your acid in your stomach. They can withstand the harsh heat. They can withstand alcohol being poured on them. They're very sturdy guys because they don't have an outer membrane to them that's very weak and requires liquid medium. Okay, so that's the big difference, which is kind of controversy and makes you think like, I thought if it has an outer membrane, it'd be stronger. But you got to think if you have that outer membrane, it's very easy for you to damage that outer membrane as well. Okay, so make sure you don't get tripped up on that because that is very important to know. Envelopes here are weak versus the naked ones. Now, with that being said, viral genetics. The way viral genetic works is this. Virus A, virus B. Okay? Now, virus A can become a whole new virus via two different ways. One, this virus with its own RNA can mutate its own RNA, and as a result, the outer capsule is now different. Okay? That's one way it can undergo. The second way it can undergo, which is the most deadly of the two, is this. This virus and this virus can infect a person. And as a result, this virus B genome is mixing with, here, let me do a different color so you can see it better. Let's do blue. This virus B's genome mixes with this virus A's genome. Okay, now as a result, the virus that develops has a mixture of both. Okay, you see how massive of a change that is? 1 versus 2. 2 has really huge change. The virus has essentially become a whole new virus. But 1, all you did was essentially do a little bit of mutation, and now it's a new type of the same virus. Does that make sense? Now, let's now start giving this names. This first one is known as a shift. No, my bad, a drift. This is a drift. This is antigenic drift. Think of it as you're drifting away from your course. Okay, so like you're a boat and then you drift off to the left. It's like a little minor change. And then you like, oh, let me recorrect. And then you correct yourself. Now, a shift is a huge change. You're like totally shifting directions. You're going a whole different direction now. Okay, so shifts are big changes. Drifts are little changes. And that's the big difference. Now, the next thing is, well, what type of viruses causes shifts versus drifts? The viruses that cause shifts are zoonotic viruses. That's why we had such a big issue with COVID, which is a zoonotic virus. It fused together with another type of its zoonotic virus inside of an animal, and it had a shift to a whole new virus. Okay? Influenza is the same issue, right? Influenza came, infected a pig. Then pig had its own zoonotic viral infection with its own pig influenza. They both mixed together, shift, swine flu. Okay? And that's how that works. So zoonotic infections are the guys that cause this. If it's human to human, you're never going to have a shift. The reason why is because humans only get infected by one type, right? They can't get infected by both. They only get infected by one type, not two different of the same category. Only A is infecting him, not A and B. Okay? Does that make sense? Okay, good. Now, here, we're just taking it on. We've already said it, antigenic drift versus antigenic shift. Right? Antigenic drift, we're just changing a little bit. Antigenic shift, we're totally different cells. Okay? Good. Now, antivirals, essentially with viruses, the way we target them is essentially in 3 different ways. And you got to think of it like this. When a virus infects a cell, there's 3 life cycle changes that the virus does. 1, it infects the cell. Right? This virus has to, one, infect it. Two, duplicate itself inside the cell. Three, release the virus outside of the cell. Okay? These are the three checkpoints that the virus has to do. So as a result, we, as knowledgeable physicians, target these three. We target entry into the cell. There's various HIV drugs out there that target entry. There's various drugs out there that target entry with influenza as well. Okay? We target the replication cycle. Yes, like the pre-exposure stuff with HIV. Exactly. The zidovudine and stuff like that. And then we target the release. If you remember that, the mechanism of action is easy because it can only target those three guys: the entry, the replication of it, or the release of the virus. It'll fall into one of those guys. Okay? Yeah, if you want to make a mnemonic for it, I'm cool with it. Just make sure after you do that mnemonic, you find out what category the drug falls into. Yes. So a fusion inhibitor would be the one that deals with entry. Exactly. For instance, for influenza, things like amantadine and rimantadine do that. Okay, these are just examples. You don't have to worry about them. You'll go over them when you get there. But things like amantadine, rimantadine, those are really big with fusion into it. Even with HIV, you got different things, right? You got trying to think off the top of my head right now. I have the name in front of my name. It'll pop up. It'll come to me again. Yeah, I'll think of it. But you with HIV, you have its different drugs. Miraviroc. There you go. Miraviroc. Okay, Miraviroc is the same kind of concept, but with HIV. Okay, good. Now, fungi and parasites. Here, the big thing with knowing of fungi and parasites is that fungi can have different formulations. So, for instance, fungi, we'll go over the cell membrane here in a minute. Fungi can become a spore, like you have in the air, or yeast. Okay? Two different categories. It's either a spore fungi or a yeast fungi. Now, the other thing you can have is what is known as a dimorphic. These are guys that are spores in one form, yeast in another, depending on temperature. An example of this is Candida. Candida albicans is a spore in the warmth and a yeast in the cold. Okay? Which is a complete flip than what other dimorphics are. Okay? But it's the same concept, a spore and a yeast, and they can change between the two of them. Now, my next question is this. Which form do you think is more deadly? The spore form or the yeast form? The yeast form is more deadly. Let me explain why. Here's the spore, right? These spores can get in various places, right? They can cause infections. I'm not downplaying that. However, yeast, if you ever see yeast, it looks kind of like this. And underneath them, they got roots digging into tissues or roots digging into bread. Okay? This here is dangerous when it comes to tissue. You do not want yeast digging into your blood vessels, yeast digging into the tissues around it, because what will end up happening is tissue damage leading to necrosis. It can even lead to abscesses. Okay? Spores, on the other hand, these guys, they can cause infections, yes. They can cause abscesses, yes. But the majority of the time, they form granulomas in various parts of the body. Okay? Yes, it's kind of like the same thing where weeds are choking off the nutrients from your rose garden. Exactly. These guys get around a blood vessel and they soak up the nutrients from it. They invade it. And as a result, you get tissue damage, necrosis, abscess formations. Exactly. Okay? So that's why yeast are very, very deadly versus the spores. Spores are deadly. I'm not downplaying it, but yeast are very deadly as well. Even more so. Okay? Now, with yeast and fungi, they have a totally different membrane to them. For instance, they got what is known as a cell wall, right? And the cell wall is formed up of what is known as beta glucan. Okay? Now, the cell membrane also has a specific type of cholesterol in it, which is only noted in fungi. Okay? And we'll see that here in a bit. But this is something that's also very unique to fungi. And these are the things we target with drugs, antifungals. Okay? Good. This here is the cholesterol, egosterol. Okay, we target this as well as the cell wall, being this glucan, the beta glucan. All right. Now, let's keep going. This part here, I want to talk a little bit about because neutrophils are very important for killing off fungi. Do not forget that. Neutrophils are very important when it comes to fungal infections. This is why Th17, Th1, this is why T cell deficiencies cause fungal to rise. Because neutrophils are very important for this process. Okay? So anybody that's neutropenic, low neutrophils, they're going to have fungal infections. And these fungal infections will be septic fungal infections. Okay? Any questions on that part of it? Yes. Exactly. They'll have recurrent yeast infections. Things like Candida, they get recurrent infections of. All right. Parasites, you're not really going to harp on these guys till later in test for your second cast. The only big thing you need to know with parasites is parasites are huge. What's the immune system that you utilize to target parasites? What's the most important cell when it comes to parasites? Eosinophils. Eosinophils. Now, B cells allow eosinophils to do its job. Okay? Because the B cells produce the IgE, which binds to the eosinophils for it to do antibody-dependent cellular cytotoxicity on the parasites. Okay? So that's why when you have a lack of B cells, you get parasitic infections. Okay? But with that being said, that's what's going on. The eosinophils are the main guy that deals with fighting off parasites, but the B cells allows the eosinophils to do its job. Let me think here. What else do I want to really harp on with parasites? Nothing else other than that. Make sure you know eosinophils. If you see high levels of eosinophils, always think this guy may have a parasitic infection, because that's a big player, is eosinophils. Now, does that mean eosinophils is not increasing anything else? No. You see high levels of eosinophils in allergic reactions, in asthma, in drug reactions. So you do see it high in other things. But if it's an infection question and you see high eosinophils, the only thing is a parasite. Okay? Good. Now, antifungals, antiparasitics. Once again, we're targeting the membrane, which is different. The cell wall, which is different. You got beta-glucan there. So those are the things we target. Are there a lot of side effects with antifungals? What do you think? Depending on which ones. Which ones do you think has a lot of side effects? Yes, amphotericin B does, because it's targeting the cholesterol. And our normal cells have cholesterol in the membrane as well, which is why amphotericin or an ergosterol these cell membrane inhibitors are the big guys that cause a lot of side effects. Okay? Cell wall ones, they don't cause a lot of side effects. Lanosterol synthesis, they don't cause a lot of side effects. Ergosterol synthesis, they can cause side effects because this 14-alpha demethylase is also known as a cytochrome P450. So what can end up happening is you start inhibiting cytochrome P450, which is very important for drug breakdown. So that's why you end up getting a lot of side effects. But outside of that, very minimal drug side effects here. Yes, drug-to-drug interactions is big here. Yep. All right. Now, the last thing is control of diseases. I don't know why it popped down so low. This is how do you prevent? So this is primary prevention. Primary prevention. Okay? For drugs or for infectious diseases. So with that being said, what do you think is a primary prevention? Washing your hands. using alcohol, rubbing alcohol, autoclaving your tools, those type of things, right? So that's essentially what we're doing. So for instance, there's two different types: sterilization, disinfection. Sterilization eliminates all forms. Disinfection eliminates many. You still have bacteria there, but a lot of them are gone. That's the difference between sterilizing something versus disinfecting something. Okay? Now, with that being said, there's different ones out there. We already talked about autoclaving. There's things like alcohol, things like ethylene oxide, hydrogen peroxide. My big thing to teach you and for you to walk away with is spore. Pay attention to the things that kill spores. Because once again, if you remember, spores are hard to kill. So you need to know things that kill them. Things like autoclaving kills spores. Things like chlorine kills spores. Things like hydrogen peroxide or iodine can be sporicidal. Okay? That's very big and important. Now, with that being said, there are other things that go along with this, right? You have different categories of risk for infection. This isn't as important because you're not really going to have a question that's like, is this patient high risk or low risk? The way they're going to do this is they're essentially going to name off different things the patient has gone through in a hospital setting. And then they're going to wonder or ask you which is most likely the root of infection. Which of these most likely caused his infection? So with that being said, high risk things most likely cause infection versus low risk things. High risk things such as surgeries, such as getting fed via a tube, parenteral fluids, IV fluids, such as anything going down or around inside your intestines, inside your bowels. Those are also very high risks areas. Lastly, that's also a big high risk thing is prosthetics. So prosthetics are things like heart prosthetics, joint prosthetics. All those things are also high risk. Okay? Good? Now, the medium thing that you need to make sure that you know is anesthesia equipment. Things like ventilators. Also high risk. Okay? Very big high risk. The other thing, things like catheters. Okay? Another anesthesia type of equipment, that's high risk. Being placed on it, being placed on a ventilator. The ventilator just sitting there isn't going to cause you infection, but being placed on a ventilator will cause an infection. Okay? All right, good. Yeah. All right. Yeah, because this is talking about autoclaving again and like all that stuff. So I'm not really going to harp on it. So that's pretty much it. We finished the whole PowerPoint because I knew we could. So I was like, let me just sit here and plow through this to finish off this PowerPoint. We went over by a little bit. I apologize on that end of it. We did take our 10-minute break, our 5-minute break. And we did start a little bit late. So it kind of evened out a little bit. Maybe we went over by like 5 minutes after all that's said and done. But yeah. So let me go ahead and stop the recording